Plasmids are autonomous self-replicating molecules of DNA, and very rarely they may be the molecules of RNA. They are not chromosomes, although they do reside inside the living cells and carry genetic information. They are not regarded as part of the cell's genome due to two reasons. First, a particular plasmid may be found in cells of different species and may move from one host species to another. That means if you consider plasmid as a part of genome, it should not occur in different types of cells and it should not move from one cell to another cell. Secondly, a plasmid may sometimes be present and sometimes absent from the cells of a particular host species. So इसी तरह सेकंड रीज़न जो है वो ये है कि कभी वो है कभी नहीं है. अगर वो किसी सेल का जीनोम है तो हमेशा सेल के अंदर उसको होना चाहिए. तो ये दो रीज़न्स है जिन वजह से हम इसे जो है वो किसी सेल का जीनोम नहीं कह सकते या उसका जो एसेंशियल पार्ट है वो नहीं कह सकते. They are not needed for cell growth and division under normal conditions. The DNA segment to be cloned is first linked to a vector DNA, which is a vehicle for carrying foreign DNA into a suitable host cell, such as the bacterium E. coli. Such carriers are called vectors. Since the mid 1970s, many vectors have been designed which mainly fall into two major classes, and these two major classes include plasmids and phages. Bacteriophage lambda and M13 are commonly used vectors and have the advantage of being able to accept large pieces of foreign DNA. They can accept the size of up to 40 kb by the bacteriophage lambda, and bacteriophage M13 can accept a piece of 10 kb. However, the most commonly used vectors for transferring foreign DNA into cells are bacterial plasmids, which accept smaller pieces of foreign DNA such as 5 to 10 kb. A typical plasmid vector is a relatively small circular DNA molecule that contains its own origin of replication, so it can replicate inside a host cell independent of the host chromosome. Although plasmids occur naturally in bacteria, where they usually confer useful properties on the cell such as antibiotic resistance, yet most plasmid vectors in use today have been specially modified or constructed for the purpose of transferring foreign DNA into the cells. Some, such as PUC18 and the blue script vectors, have been engineered to make many hundreds of copies of themselves and any foreign DNA they contain once they are introduced into a host cell. This process of producing a large quantity of identical copies of any chosen DNA is referred as cloning. This process is called cloning, and the cells derived from a host cell containing a recombinant plasmid is referred as cell clone. For cloning to work, the vector must have a cutting site for a restriction enzyme that makes it possible to insert the desired fragment of DNA. ये एक प्रॉपर्टी है जो इसमें होनी चाहिए. और सेकंड प्रॉपर्टी, the vector must be selectable so that cells that have taken up a vector can be identified and can be separated from those cells who have not taken up that plasmid. To overcome the difficulty of inserting fragments into vectors, most plasmids have been constructed with polylinkers or multiple cloning sites. What is this multiple cloning site? This is a piece of DNA that contains one or more unique restriction enzyme recognition sites. Moreover, most plasmids contain antibiotic resistance genes or other selectable markers that enable cells to be isolated from cells that have not taken up a plasmid. In the early years of cloning era, Boyer and his colleagues developed a set of very popular vectors known as the PBR plasmid series. Nowadays, one can choose from many plasmid cloning vectors besides the PBR plasmids. One useful, though somewhat dated, class of plasmids is the PUC series. One of the most notable plasmids, termed PBR302 after its developer Bolivar and Rodriguez, was widely adopted, and it illustrates its desirable features of a cloning vector, which are given below. The plasmid is much smaller than a natural plasmid. This PBR322 is much smaller than normal plasmids, which makes it more resistant to damage by shearing and increases the efficiency of uptake by bacteria. This uptake of DNA is termed as transformation. A bacterial origin of DNA replication ensures that plasmid will be replicated by the host cell. Most plasmids, including PBR322, have a relaxed origin of replication, and which means replication is not tightly linked to the division of the cell. It can replicate independent of the replication of chromosome or replication of the cell. The genes coding for resistance to antibiotics have been introduced, which allow the selection of cells which contain plasmids. There are single restriction recognition sites for a number of restriction enzymes at various points around the plasmid, which can be used to open or linearize the सर्कुलर प्लाज्मिड. सो इस प्लाज्मिड के ऊपर जो हैं वो सिंगल रिकग्निशन साइट्स या सिंगल रेस्ट्रिक्शन साइट्स जो हैं वो मौजूद हैं, और इन साइट्स की वजह से हम इनको जो है वो लीनियराइज कर सकते हैं, ओपन कर सकते हैं. सो दिस इज़ द PBR322. So here you can see this is the origin of replication, and this is ampicillin resistance, antibiotic resistance portion, and this portion contains a number of restriction sites for different restriction enzymes such as EcoR1, HIND3, EcoR5, BAM, H1, etc. So this is PBR322.